they're fascinating and they're super useful, but also they lead us, I think, somewhat surprisingly, at least to the field at the time, to the development of a very different kind of algorithm, namely combinatorially precise exact algorithms for solving structure. And so, and that comes from understanding the, essentially the physics in some sense. So let's talk about RDCs. I'll show you some slides later for things that are easier to visualize. As always, if you have a question, feel free to ask. And if you're shy, you can use the chat, in which case probably Andy will see it and interrupt me because it's hard to see the chat when I'm screen sharing. All right, so let's talk about what we're trying to do here. I'll write some stuff down and I'll show some slides later. By the way, I appreciate the discussion on Piazza. We had a very good and robust discussion. I'm proud of you all for asking these questions. The level of understanding and engagement, I think, has exceeded almost any other version of this class, so great job. And we'll try to get at all the things you're intellectually curious about. So I want to start by going back to talking about what's the basis of RDCs, like why do they happen, and also what does an experiment look like that would measure these things for the experimentalists in particular. But we're interested mostly in the information content here. So let's look at a couple of examples that I alluded to before. So let's look at something like this, say from essentially organic chemistry. So we have a methylene carbon. And let's compare that to a methine. And these are carbon 13 here. And basically the difference between these things is going to be that for the methylene carbon, we're going to have 2 protons attached to the C13 nucleus. Here we have 1 proton attached. And if you record a one-dimensional NMR spectrum here, you can see the effect of the attached proton resonances on the C13 resonance. So roughly speaking, and I'm not a great artist, this one will sort of have three peaks here, right? Something like that. And this one here will have something like 2 peaks here, right? And again, the physical basis for this is the energy levels of the different spins. And you've probably seen some pictures like this in your readings. I know you've been asking questions. The energies, we have 2 different levels here for the proton spin and 2 different levels in some sense for the carbon spin. And so what happens is that, and you only can be in these up and down states. Now, what are these things exactly? So of course, this corresponds to essentially a fairly complex object. You're talking about in the up state, we're talking about a spin that's going to precess around the magnetic axis here. So it's going to, say, precess in this direction like this, rotating around this thing like that. And that's say the up spin. And in quantum mechanics, you might have seen this represented by this kind of notation, a bracket and 1. And then of course the down spin has this vector exactly opposite, pointing down instead. And that would be the 0 state in some sense. I'm simplifying here. And then the actual state of this is in any sense, is a superposition of these 2 things. So we have 2 coefficients here, alpha and beta. We sum the alpha and beta, actually are complex numbers. And they're represented by essentially the wave function, right? So this is a very complicated object, but its basis, in some sense, the things that make it up are these up and down spins. So the basic way we can think about it in this kind of pre-theoretical way that we're developing this material is that in this case here, again, we're looking at the C13 spectrums. This is omega C13. This is omega C13. Is that the C13 resonance is split by having the attached proton here into 2. And here it's split into 3 because of 2 attached protons. So the key thing here is that, a way to understand this is by these energy levels and these up and down spins, but it's not something that can be completely explained just using spinning magnets in some sense. You need to have this sort of quantum mechanical view that you have these different discrete states in some sense. And what's also important is if you do an experiment to manipulate and adjust these states, that it will have similar effect in other experiments where you use the same interventions. So let's be a little less mysterious about this and try to show some more examples of what's going on. And the example that I want to do, now that we sort of understand these splittings a little bit, which are incredibly important in organic chemistry, is to look at how we would actually start to develop our experiments here. So let's go back to our friend, the HSQC spectrum. So remember, you have a protein like this. I'm going to draw a cartoon protein that looks something like this. And, along this backbone, you're going to have bond vectors that are amide proton bond vectors like this. And so roughly speaking, an amide bond vector will go from a nucleus A to a nucleus B like this. And in our case, we're going to have A is equal to an N15 nitrogen, and B is going to be equal to a proton, actually the amide proton like that. All familiar kind of notation. We have different ones, roughly one per residue, with the exception of prolines and the C-terminus. Okay, so what we do here in this HSQC experiment, right, is we observe a peak or a spot for each one of these moieties. And here we have the frequency of the amide proton. And here we have the frequency of the N15 nitrogen it's attached to. And we hope to see a peak for each one of these. And they have different frequencies because you have dispersion due to the differences in local electronic environment. So the question here I'd like to motivate is why do we just see one peak? Why aren't these split? And indeed, they would be split if you use the same kind of experiments that are used in organic chemistry. In fact, what you would see for each of these peaks, let's zero in on this thing here. What you'd see if you did these without some extra fancy pulse sequences is you would actually see something that looked more like this. You would see something along these lines, and that's exactly what you would expect because in some sense the two levels of the N15 nucleus split the proton, and the proton splits the N15 resonance too. So you actually would just as in these cases with these organic chemistry things like methylene, methane, you actually would see 4 peaks. Now, if you have a big protein, and we've worked with proteins by NMR that have up to 650 residues, then you'd have 4 times 649 residues, spots there. You have a lot of spots. And so one of the things you do in biomolecular NMR as opposed to organic chemistry is you decouple these. And there are a couple of ways you can decouple. Actually, 3 different ways. One is you can kind of average these peaks. The other is you can explicitly make one decohere by sort of shining an RF flashlight at it, making it turn off. There are various tricks you could do. And so that what you see in the NMR case is basically you see a peak that's the average N15 HSQC like this. So this basically can be thought of as the primary thing it's doing is doing a 90-degree pulse to transfer magnetization from the proton to the N15 resonance, right? That's the primary thing. And so that primary, primary is an informal term, but the thing it's doing is using these operators, which you can think of as these pulse sequences that move the magnetization axis. But you can think of these as operators like, tipping things around the z-axis for the proton, followed by tipping things around the z-axis for the nitrogen like this. This is sort of this is the sequence of operators. These are actually, matrices that are multiplied together in some sense. But I want you to think about this as the sort of quantum mechanical type of this experiment. In other words, it's manipulating the 2 spins by doing this. And so if I do another experiment with the same type, I'm going to see similar kinds of I'm going to see similar kinds of interactions. And a sort of key thing here is that these differences are called the scalar couplings. This is the 1J NH scalar coupling. So it's a coupling across just one bond vector here from the N15 to the NH. So if we're going to do something to the system, we'll observe those splittings in different experiments. So this is way of sort of typing the information that we're going to get here. Now, let's take sort of a geometric view and say, how does this relate to RDCs in some sense? I'm going to pause for a moment to plug in my power cable, which I think would be a good idea to make sure we don't lose signal here. All right. Okay, so let's look at our picture again. Here, I'll just put a little piece of this here so we can kind of get oriented. So we're interested in, the structure of a protein like this, right? And we have these amide bond vectors that are different orientations. That's the key thing. So this is the picture I'm going to blow up to talk about, to explain how RDCs work. All right. So one way to think of this is to draw a little picture like this. At an instant time, this is our external magnetic field. I use sort of physics vector notation. And we have a bond vector here, which I'll call V, like this. And there's an angle between these things, which I'll call theta. Okay. Now, and of course, we have two nuclei here, A and B. And as before, I'm going to have A, in this case, be my N15 nucleus, and B be my amide proton. Like that. So this is my amide bond vector. This could be different orientations here. Now, the dipolar coupling between these things here is equal to the following quantity. And I'll parse these symbols for you. To B, to H bar. All right, so this is an intimidating equation at first, but we're going to try to make sense of it and then use it. So what are all these terms? Mu0 is the magnetic permittivity. It's like how well does magnetization transfer across media like water with solvent, things like that. Gamma alpha and gamma A and gamma B are the gyromagnetic ratios of the two nuclei, N15 and HN. There's no classical equivalent of these. You can think of these as kind of how loud the nucleus screams, or sort of roughly equivalent of sort of inertia in some sense, how hard it is to get them to do something if you hit them with an RF pulse. Smaller things typically have much larger gyromagnetic ratios. So electrons, much better, much higher than protons, which are much higher than heavy nuclei. Constant pi, RAB is the length of the vector. So if I have my vector between these things, it's just the magnitude of that vector. And then we have this strange thing here. Now you see it's dependent on this angle here, which is kind of nice. Where does this sort of come from in some sense? Well, this is the second Legendre polynomial of cos theta. And if you've studied some physics, which I don't expect you have, it comes from the spherical harmonics that are used as a basis for things that live on a curved surface like a sphere. And then finally over here, I want to mention we have these operators, which are the basically quantum mechanical operators that are selecting for how we get this value. And just to show you, it has the same, again, it has the same quantum mechanical type as the HSQC, right? So I've just changed the names here. It's this operator followed by this one. You're flipping this along the z-axis here. And all this means for us is that if we do these operators, this effect will appear in an experiment that has the same operator. So we should be able to see this actually in an HSQC experiment. And how does that actually going to work? I mean, how's that going to work here? Let me continue to parse that as I talk about what we'll actually see. The brackets indicate averaging. So this is a time and ensemble average. So you have an ensemble of billions of proteins in your solution, pretty concentrated solution. And you're averaging over time, usually a matter of seconds or milliseconds. And a fairly deep assumption in this kind of physics, which is basically looking at a statistical mechanical readout of quantum mechanical manipulations. The ergodic hypothesis, this is ergodic, basically says that the time average and the ensemble average should be the same. Now, let's parse this a little more, like what's going on. What would you actually observe here? So why should we see anything in the HSQC experiment appropriately modified? And the answer is that we really don't in the case where we decouple over here. And that's because we've averaged everything out. But basically, the way this experiment is set up is that if you do these manipulations, the splittings are measuring this 1J&H scalar coupling through one bond. So this difference here is measuring those couplings. And so the trick I'm going to use is I'm going to turn off the decoupling just in the N15 dimension. So I'm going to have splitting in the vertical N15 dimension, but not in the proton dimension. And so my spectrum is going to look a little bit different. So whereas before, I had spots that looked like this, right? This is my, again, omega frequency of 15. I had spots that look like this because the averaging. And now, before doing the experiment, I turn off the decoupling in the N15 dimension, and I get this instead, and the blue point average is not visible at all. And now, this distance here, this difference in frequency, basically, this measures the splitting on J and H, right? This measures this thing here. So this is pretty cool that I can measure these splittings. So the main thing is that the dipolar coupling here is going to contribute to that difference, contribute to that size. Okay, that's pretty interesting. However, if we look at a protein in solution, right, this thing is tumbling rapidly. The average reorientation time for a small protein is about 10 nanoseconds, for a bigger one, maybe 20, 30 nanoseconds. So these things are spinning and tumbling rapidly and isotropically. And all that matters is orientation, and that's averaging out. So in a solution NMR experiment where everything is nicely averaged, these couplings actually average out to zero. And that's why before, when I wrote down the Hamiltonian for a typical protein experiment, we had the Zeeman potential, we have the chemical shift potential that determines, very big actually, determines what frequency each nucleus has. Then we have the dipole-dipole interaction. And then later on, way down here, we might have the NOE if we use the appropriate pulse sequences for distances. But the main thing is that in solution, so Ln for solution, outright isotropic, meaning the same in all directions, this actually averages out to zero. And so I've done all this math in order to add something to my Hamiltonian that's going to average to zero. And even if I use particular pulse sequences to select to read this, this contribution is also going to be zero. So these shifts, these differences here are going to be not affected by this. In other words, there's a process that's making this splitting happen, just as there's a process that's making these splittings happen, right? There's a process making these splittings happen. A contribution to that splitting is the dipole-dipole interaction, and here's the equation for it. But the problem is it goes to zero, which is, not a lot of work for nothing. And so what a bunch of people figured out, this comes from the liquid crystalline world in some sense, but really Ad-Bax at NIH and James Prestegard at the University of Georgia figured out that instead of this tumbling, rapidly and isotropically in solution, what you could do is you could prepare a special biochemical bath in which you have the protein, but you titrate in media that partially align it. And what's the intuition for this? Well, in solid state NMR, where everything is aligned, everything's basically a crystal, these are not zero because everything is aligned. And in fact, they're enormous. And so to give you an idea, they can be like 20 or 30,000 hertz. whereas the chemical shift part is maybe 200, 300, 400 hertz. So they're actually the largest dominant thing, and they're actually not used directly in solid state anymore, NMR, because they're so huge. So if you just aligned everything, you'll see the signal, but it will swamp everything else and be enormous, and you can't resolve it. So the idea is you have an aligning medium, and you might use like phage or bicell. So you put in these big things like this, and you titrate these in until the molecule is partially aligned. And the way of thinking about this is the molecule is tumbling freely and isotropically, as I'm illustrating here. But as you titrate these things in, the idea is that if 1 in 20,000 times, it encounters either steric or charge hindrance from these big walls you're titrating in, then what you're doing is you're biasing the equilibrium distribution of orientations. So all of a sudden, it's not tumbling isotropically, and you have an anisotropic bias. In other words, you have an actual distribution that's not uniform over rotation space. And then you can experimentally see these signals coming back. So you actually titrate these in, you have rules of thumb, and you titrate these in until the RDCs are about between plus or minus 30 hertz typically, or plus or minus 20 hertz. So a small signal, but highly significant. And there are lots of ways of doing this. Aside from titrating in phage or bicells, there are actually 50 or 60 different aligning media. It's a biochemist's paradise. One really cute thing you could do is actually put these in gels, because proteins tumble in gels. There's plenty of water in there. If you then stretch or compress the gel, then you get an alignment of the protein, because you start to add stress to the medium. So you can do your NMR measurements in a gel with lots of water. The molecules for proteins tumble rapidly and isotropically. Then you stretch or compress your gel, and you get an aligned condition in there. This was actually pioneered at NC State. So a very clever idea. So many different aligning media. It's a completely experimental process on how you get these to be aligned. But once you get partial alignment, which is why these are called residual dipolar couplings, this signal starts to come back and it becomes non-zero, right? And you actually start to get a measurement. So how do we interpret this measurement? What do we think about it? Well, again, we have these averages in some sense. So let me give you a hint about how to interpret this. So there are all these constants in here, right? And to a physicist, these are super important. But to a mathematician, these constants all just are like K or something, because they're all always the same. So that's kind of one difference in perspective on how to think about this. Second, if we're looking at an RDC and a bond vector whose distance between the nuclei we know, and we definitely know how long an MI bond vector is for many crystal structures, about 1.8 angstroms, this becomes a constant. So this entire first term here becomes a constant because we're not actually measuring the distance, we're using it as a part of the model. So we model in this term here as an average. Now, as for this average here, which of course goes to 0 in an isotropic, rapidly tumbling solution, this thing here now will start to come back and give us orientational information about what's going on here. And one way to think about what this term actually is it's basically proportional, or signal will be proportional to the square of the dot product. And that makes sense, right? So if you look at the dot product of the external magnetic field and our vector V, this is proportional to this quantity squared. That's pretty cool. So what we're doing is we're measuring basically orientations. We're measuring in one shot the dot product squared of each bond vector with the external magnetic field. And one of the things that's super cool about this is that it's our first NMR measurement in this class, which is in a global coordinate frame. So if you have a protein like this, you do NOEs, just like a surveyor trying to survey, forests in Oregon or the Mississippi River, these little tiny NOE measurements you're making, these little triangulations and things, error can add up as you go through making these measurements. And by the time you get to the other side of the protein, you have accumulated error, and the domain way over here might have a totally different orientation in your model, which is incorrect. The beautiful thing about RDCs is that this bond vector here, this one here, and this one here, you're measuring their orientation relative to the same external huge magnetic field, right? And so you're measuring the dot product of V1 with this, V2 with this, V3 with this, all in the same global coordinate system. So RDCs become much more like data in crystallography or cryo-EM. They're globally referenced. And even if this was a megadalton protein, which you wouldn't have, but you might have a complex, if you had a very large protein, and these things are tens of angstroms apart, they're still globally referenced, i.e. you can look at the dot product of this and that, like V1 and B0, V2 and B0, and then you know something about the relative orientation of V1 and V2 because they're in a global coordinate frame. So this is very exciting because all of a sudden our quality of structures and our ability to globally reference goes way up. But this is still a daunting equation. So let's try to come up with some intuition for it. So one thing we could do is we can collect all the constants, and we can kind of write this just as K, a constant, times this averaging here. And one way to write this that's very popular is called the so-called alignment tensor. You can actually rewrite this as V S V T, like this. All right. So this is pretty interesting. What is this thing? So this is our constant. That's the constant that incorporates all of this stuff here. This is our bond vector. And then we have this weird matrix here. So let me tell you some things about this matrix. So first of all, this is a 3x3 matrix, and it has real values. And of course,
Bruce,
these things, yes.
Sorry to cut in, but shouldn't it be T transpose B transpose SV?
Yeah, sorry. Thank you. Please watch me carefully. Thank you, Andy. Speed transpose V. So each of these V's, of course, is a this is a vector, form X, Y, Z. Like that. So this multiplication makes sense. So let me just kind of set this in context by talking about kind of how physicists think about this kind of thing. If I have one of these 3x3 matrices, let's just take the identity matrix like this, and I go for two vectors V and W, this just corresponds to the inner product, right? And this is the 3x3 matrix. And more generally, you could have another symmetric matrix there instead, and that would give you a different construct. It would give you a different metric that changed from point to point. So this isn't such a strange beast here. It's kind of a strange sort of inner product here. And it turns out that this matrix, again, 3x3 in real value, it's traceless. And that means if you add up the elements in the diagonal, they're 0, and it's symmetric across the diagonal. So a lot of structure about these things here. So this is a special way to write it, and kind of a much more compact way of thinking about this strange equation here. So in some sense, the matrix here and this juxtaposition, so to speak, of the vectors, the matrix is going to encapsulate sort of the average orientation of the system, which is what we're really trying to get at here. So let's go in a little more detail about this in some sense. All right. So again, key points here before I go on. This is all a constant if we know the bond length or model the bond length. This is an average time and ensemble. It's proportional to the square of the dot product of the bond vector and B0, the magnetic field. We can, in general, write inner products like this. This is with the identity matrix. This is the identity matrix, i.e. 1, 1, 1, like that. But in general, it can be something more complicated. This is kind of a generalization of the dot product. It's kind of like energy in some sense. And this matrix here called the alignment tensor, or the SOAPy matrix. This is going to encode our orientation, and this is something called the alignment tensor. So it's called the SOAP matrix because it's a 3x3 matrix with these properties, and alignment tensor because you can multiply the velocity, the vectors like this, and it's more or less quadratically dependent upon the vector V and linearly dependent on the coefficients within S. So because it's quadratically dependent on the entries in your vector, it's going to be exquisitely sensitive to changes in geometry. And Andy, thank you for finding the typo. This is complicated stuff, so I definitely want to get this correct. All right. So this is an intimidating object, and I want to give some intuition. What's our question?
Yeah, sorry to interrupt. Can I ask a quick clarification question about the scalar? So are you saying that the reason that when you when you're not averaging over, when you're averaging over all of the rapidly tumbling proteins in an isotropic material, you're gonna see only you're gonna see the decoupled signal, because like you're gonna average to the middle. Is that what?
No, the decoupling and the zeroness of those are actually two different phenomena. So the decoupling is a different question, and it devolves to whether or not the attached spins split a central spin, right? And we're not going to get around that. There is a process to get rid of that, but that's not part of this. So this process is not called decoupling, it's just called averaging. So basically, so that doesn't collapse anything here. You're still going to see two different peaks here, the two red peaks. But I guess one way I can explain this is as follows. So here's the spectrum unaligned. See, unaligned spectrum would look like this. And the claim is that when this term is averaging to zero, it just doesn't contribute to the difference here, the length of this red line. But if we look at the aligned spectrum, it'll look like this. So I'll draw them next to each other, but they're really right on top. So instead of this, we're going to see something like this, right? And here we might see something like this. And here we might see something like this. So the RDC is the difference between the green line segment and the red one. So when it's unaligned, there's no contribution. This doesn't make the splitting, the 1JNH splitting, either bigger or smaller. But once you align it, this signal comes back and it's added to this. Why is it added? Because it has the same quantum mechanical type as the experiments that measure the 1JNH splitting. And so in short, if you do this experiment in the unaligned setting where you have rapid and isotropic tumbling, what you see, I know this figure is messy, what you see are just these 6 dots, right? Corresponding to the decoupled HSQC spectrum. When you align the system, then you see, instead of that, these 6 green dots like this. And then you subtract the length of the green line segment from the length of the red line segment, and you get your RDC. So in short, your two spectra aligned and unaligned would look like this, if I can draw them. So this is unaligned and aligned. So here you would see just this. I'll try to draw it to scale. And your aligned version, and we know we can, I think it's fair for me to put in the line between them. The aligned version, these change, they get bigger or smaller. So this one gets like really big, this one gets, tiny, and this one gets really big like this, right? And so in computing the RDC, what you do is you take the difference of this length from that length, this length from that length, this length from that length. So what you're doing is you're changing the size of these splittings. Does that make sense?
Yes, thank you.
Yeah. So it is confusing. Decoupling is a different phenomenon from rotational averaging. Great question. Any more questions before I go on? All right. So I want to make this a little easier by talking about a connection to how computationalists think about these problems, which is principal component analysis, which this is similar to. It's kind of a physical process for principal component analysis. So suppose I have some distribution of points in 3 space, and it's 0 mean. Mean of this is 0. And call this P in R3. And for now, it's a discrete finite set, right? So in some sense, PCA, this is an approximation for intuition, PCA of P returns the best fit ellipsoid right? This is pretty interesting. So, canonical picture of this, you have a bunch of points like this, say, and you might have some outliers, right? These are your set of points P that from some data, right? And I've just drawn R2 here, our dimension reduced picture. And then in this case, E might look like this, right? So pretty amazing you can do that. Classic mainstay of dimension reduction. Not really machine learning, but it's used in machine learning, right? So how do we represent E? Well, you can represent E. I mean, E has 3 axes, 2 in this case, right? That are orthogonal, right? And the axes of this guy can be represented by a rotation matrix, right? Basically define a coordinate frame. So it's an orthogonal right-handed coordinate system. And then 2 scalars will suffice to include the stretching, how the axes are stretched. So of course I've got this thing here, but basically the idea is, let me draw my ellipse again like this, as well as I can like this. Right, so this thing is both rotated, based from the coordinate system here, my natural coordinate system, and stretched. So what I'm going to say is, well, it has a major and minor axis, just like we learned in high school, like this, right? And the difference between these axes and these axes, this is just a rotation, right? And here a 2D rotation, but in general a 3D rotation in this case. And then of course I've stretched and squashed it. So let me just arbitrarily say that the stretching here is 1 and the stretching here is lambda 1, right? And so if the rotation is called R, a rotation matrix in some sense, and by the way, rotations are covered in CS 230, but I'll help you learn about them. So you can define the ellipsoid by triple, the rotation, and lambda 1 and lambda 2. Here I've let one of the eigenvalues be 1, but basically that defines how you stretched it. So all I need to define the ellipsoid is I start out with sort of a canonical circle, I rotate it, and then I stretch it. And these guys, by the way, are called the eigenvalues. So this is a first order approximation, saying P is represented by an ellipsoid, right? This is like not necessarily correct, but it might be a good approximation. So the P to an ellipsoid is completely a first order approximation, but the representation of an ellipsoid by R, lambda 1, and lambda 2, this is exact. So let's just write that down. So when we say, P is represented by the ellipsoid, which is PCA of P, this is a first order approximation. And you can see that, right? It's not perfect because I got outliers and stuff like that, right? But the representation of E as the triple R lambda 1 and lambda 2, this is exact. This transformation is exact. Okay, so this is pretty cool that I could do this. Now let's apply it to something kind of molecular. So suppose we have a molecule that's approximately aligned to be 0, right? Because we used an aligning medium. So let me draw something a little bit more interesting than what we had before here. I've got some helices here. I'll draw them like this. And how about one in the back? Okay. And let's hook these up in some fashion like that. And hook them up like that, right? We have something coming like that. So we have a molecule, it's approximately aligned to B0, right? So we have an external magnetic field like that, which is from the NMR magnet. So, the ergodic hypothesis, again, is going to say that we're going to look at a time or ensemble average, right? And they're going to be the same, time or ensemble, like this. So one way to think about it is, suppose we fix a frame on the molecule. So this is a PDB structure. If you download something to PDB, it is in some orientation. If we align the molecule, align the protein, that's rotating that frame. So we imagine that in this coordinate system, we now have a coordinate frame, right-handed coordinate system. Like we fix a frame on here like this. Let's call that frame F. So right-handed coordinate system, I hat, J hat, K hat like this. So basically, as this ensemble wiggles, right, over time or across members of the ensemble, then if we can either view the frame, here's my right-hand rule, I've got my frame, the frame is wiggling like this a little bit, right? As the ensemble moves around like this. The other thing we can do is we can move to the frame of the molecule. So now the molecule is like a bucking bronco. I'm riding on its back like I would a horse. And as the horse is bucking around, I can observe the world. So now one thing I can observe is this flagpole. The flagpole happens to be B0. And I can, as I ride the back of the horse like this, bouncing around, I see the flagpole, and the flagpole is waving around, right? And the flagpole isn't changing length, so I just view the end of this. So basically, if I'm in the molecular frame riding the bucking bronco, which is my protein here, I'm observing the tip of the flagpole, which is lying obviously on a sphere because it doesn't change in length. And there's a distribution of observed points of the flagpole tip. So I can treat this distribution of points representing the basically orientations of the external magnetic field as viewed in the frame of the molecule as a distribution like P. So I can then take an ellipsoid to represent those and represent that by a matrix. So that's the idea. So the basically conceptual steps here are put a frame on the protein. This is simple because PDB has a frame, right? Here we go. And then what I could do is like in physics, I could pass to any inertial frame. So I'd have pass to F. Now I'm observing riding on the back of the protein. And then you can say in F, observe basically B0, which is waving around. And think of it as a function of T. I'm only interested in the endpoint because, you know, it's not changing size. And then as I observe it, I get an observed set of points P, which are the set of orientations of the molecular frame. I now take P, I do PCA on that, I get an ellipsoid. I can represent the ellipsoid as a matrix and two eigenvalues. In fact, 3 here because it's 3D. And this is an exact representation of the ellipsoid. So this is how a matrix like this, like the SOAP tensor, can represent this averaging. Conceptually, it's actually a little bit different, but it's close to this because the matrix represents the rotations that I'll see on average, and the eigenvalues will represent the stretching. So this shows, conceptually at least, how I can observe, you know, the experiment observes the orientations of B0, the flagpole sitting outside as I ride on the bucking horse. And then what I'm going to do is I'm going to fit an ellipsoid to that. And then I'm going to represent that ellipsoid by a matrix. And this corresponds approximately to the SOAP matrix. That's the intuition behind the SOAP matrix. Okay. Now, what about algorithms? How can we actually, you know, think about how to do this? And to do that, we need some intuition here from probably one of the most useful algorithms in computer science, namely the singular value decomposition. Now, I recommend highly Carlo Tomasi's book on this. Let's start a piece of paper since I can't spell. Singular value decomposition. I'm only going to tell you a little about this. There's plenty in the textbook. But basically, the idea is if A is a real valued matrix, which is M by N, then you can compute U and V, which are orthogonal matrices, such that A is decomposed as U transpose sigma V, where sigma is a diagonal matrix. And its diagonals are called the eigenvalues. And yes, SVD can be used to compute PCA. And furthermore, if A is symmetric, implies that U equals V, which is pretty amazing. So if we look back at our situation here, we have this matrix here. It's 3 by 3, it's real valued, it's traceless, it's symmetric. That's what we're going to use here. Certainly it's real valued. So we can just decompose this in a very nice way using SVD. So therefore we can conclude that S is equal to U transpose sigma U. And these are the eigenvalues, these diagonals, this diagonal matrix here. and they define the stretching. And this is a rotation matrix, 3x3 rotation matrix. So it rotates vectors and it defines the frame. And this is our alignment tensor. So the alignment tensor can be thought of as defining the B0 distribution in the molecular frame. Or if we want to be in the laboratory frame, it defines the molecular frame average in laboratory frame. But I kind of like this one because it's closer to PCA. Okay, so this is really beautiful. And again, what does it mean to be a rotation matrix? We have to be, it has to have orthogonal rows and columns, and it has to have determinant 1. So it'll rotate anything. Now, what I want to do with that information is just show you one application, one complication, and then this leads directly to exact algorithms now that we understand the physics a little bit. And I want to show you some slides in a few minutes to show you what these kind of curves and things look like, because they're much easier to understand than by drawing them, but I'll do a little bit. So let's look at several applications. SVD is incredibly useful. I can use it to do PCA. I can use it to solve minimization problems. I can use it for all sorts of different things. But I can also use it empirically to compute a SOAP matrix. And this is incredibly useful. And by the way, before James Prestegard and his postdoc discovered this, people were using Monte Carlo algorithms and simulated annealing to compute it. But it turns out you can compute it exactly in closed form, which is pretty remarkable. So suppose we have a protein. Again, my protein's going to look a bit like this, right? So my protein looks like this, I know. And basically from modeling or from experiments, like I can look at CD, or I can look at just the chemical shifts, or I can look at predictions. I know it has some helices in it, at least one helix, and I know roughly where the helix is. Or if you like to stay with the NMR world, I could just look at the phi bond angle from the J3HNHA experiment, see that it's the alpha helical region. That plus the chemical shift should tell me where the helices are. So I want to be able to identify a helix in my protein, even though I don't know its three-dimensional structure. So suppose I know that it has a helix, and suppose I'm going to say, well, I don't know the exact geometry of a helix, and I realize they can be bent a little bit, but it's probably okay, I'm going to say, to assume an ideal helix. And one reason this might be reasonable is that if I look at a dipolar coupling here, this is an R, and I, again, use what Andy says, this is V transpose SV. Remember, alignment tensor, here I have 2 times the bond vector. I have a bond vector like this is V, and here's my experimental reading. So the thing to notice here is that the value you measure is quadratically sensitive to changes in the bond vector orientation. It's exquisitely sensitive to geometry changes, but it's only linearly dependent on the alignment tensor. So maybe I can fit something approximate. So the trick here that works pretty well is to say, well, I'm going to just assume I know it's a helix from experiments, other experiments. I don't know what the helix looks like. I'll just fit an ideal helix and use that to bootstrap my knowledge of the alignment tensor, because obviously you need both the alignment tensor and the measurements to solve for things. I mean, use the inverse problem. What I've done in an actual experiment is I've measured R, and I want to recover the geometry here, which is the bond vector orientation. But without knowing S, that's rather difficult. So what I can do is I can use an ideal helix to bootstrap that. Once I have bootstrapped and I know something about S, then I can start to approximate these vectors. So the idea is I take an ideal helix like this, or I could use a homology model like this. And from the ideal helix, I have various bond vector orientations, which I'll represent like this. So here I'll have V1, V2, V3, up you'll have V4. Just take it from the ideal helix model like this. And then I have a sequence. The trick here is I want to use SVD to solve a linear equation of the form A X equals B, where A is a matrix, X is the unknown, and B is a known vector. And I'm not going to set this entirely up for you, but I'll give you the idea, is that suppose in this situation here, I've measured a bunch of dipolar couplings, and I know a bunch of bond vectors. Then I can set up a system that looks like this. D1 equals V1 transpose SV1. D2 equals V2 transpose SV2, all the way down to Dn equals V2 transpose S, Dn transpose SVn. So I have a system of equations like this. Again, think of what I have here. These are my measured RDCs. These are my vectors, bond vectors, and they come from my ideal helix. And S is my unknown. And so I can transform this into a system like this, and SVD will give me the optimal least squares solution to it, and it runs in polynomial time. And in this way of thinking about it, my unknown X, just for fun, looks something like this. Let's look at our alignment tensor, right? So our alignment tensor is traceless and symmetric. So it means that the entries here, SXX, SYY, this one will be known because it has to sum to these things. So I have SZZ. So I have to have SXX plus SYY plus SZZ equals 0. This is what it means to be traceless. So I don't need to include that one. And then I have these entries here. So I have an entry here, which I'll just call this W1, then I have W2, W3. So the only entries that I need to have are these 5, because this one's determined by those 2, and these are symmetric over here. So my vector X is just SXX, SYY, W1, W2, W3 transpose. So it's just these entries here vectorized in this format. And then it's an exercise to figure out what B is and what A is. Happy to show you how to do that. So SVD can be used to do this. So basically in this workflow here, we start with the experimental measurements D1 to Dn. We've measured a bunch of RDCs. We start with an ideal helix. And that gives us our VIs, our vectors from the ideal helix. We put these into SVD, and we get out our alignment tensor, or at least an approximation. Now, having done the helix, suppose we go back to our model. So now we have not only B0, but we have the alignment tensor. Now, when we measure an RDC at some strange part of the protein like this on vector, say, let's call this vector G. Now I can interpret what that means if I've recorded RDC, say R sub G, for this vector, which is called vector G transpose SG. I now know this because I measured it. I know this because I estimated from an ideal helix. And now I have the problem of trying to solve, and I want to say, what is GT? Or what is G? Equivalently, what is G? Just transpose each other. So again, I have an equation with just one unknown, although it's a vector unknown. I know R of G from experimental measurements. I estimated this by bootstrapping from a helix. And now I have these two guys, same base, same guy base. If I want to estimate those, I have to invert this. And the question is, what does the geometry of that look like? And that's what I want to show you some slides on in a couple of minutes. Before we take a break and I set up the slides, I want to just show you that it's slightly more complicated than this, just a little bit. And this is covered in the textbook, but I just want to give you an idea of the complexity. So I did say before that we had this marvelous idea, which is we put a frame on the molecule. We now move to the molecular frame. In the molecular frame, the experiment observes essentially the cloud of points B0 as a function of time, although you don't see the time. All these different points, which are how the flagpole looks when you're riding on the horse that's bucking around. And then we just do PCA on that to fit an ellipsoid, represent the ellipsoid by a matrix and the eigenvalues, which is an approximation to the SOAP alignment tensor. That's pretty cool. one complication, and comes up a lot in this work, and this is from the textbook, is that if you look at our RDC equation, which I had here, you'll see that you have this cosine squared. And this roughly means that if I have an alignment tensor, or I know B0, and I've measured the RDC, there's basically two solutions. Like in robotics, there's an elbow up and an elbow down solution. So we can't distinguish that solution from this one on that information alone, right? Because this also has the right angle, the dot product is going to be fine. So the thing is that you basically, because of the square, you're going to have two solutions, just as you do with polynomials. Physicists call this as something that's because of the symmetry of the dipolar operator. From our point of view, it's because polynomials that have a square in them are going to have two solutions. As roboticists, we can see that kinematically you have a up solution and a down solution for these two things. So what it means for our actual fit is that our real situation is something like this, which of course is appropriate to cover in a textbook, but in a lecture like this is a little complicated, is that we do have a cloud of points and we take PCA. However, we actually have two clouds, up solutions and down solutions. And so our PCA is a very broad approximation. It's an ellipsoid that fits both of these like this. And again, you can read the textbook for more details on that. But nevertheless, this works quite well empirically, and it's a good approximation. So this having been done, what I'd like to do is take like a 2-minute break. I'm going to make myself another cup of tea. And then I'm going to set up some slides, and I want to show you some pictures of this. The pictures will be, what they'll be trying to do is get at the algorithms. Now we've seen SVD and PCA, that's pretty cool. Well, what about this question here? When we've measured the dipolar coupling here from experiments, we've estimated the alignment tensor here by bootstrapping from a ideal helix geometry that gave us all the VIs. Then we use that ideal helix geometry to compute an approximate alignment tensor. So now we know this guy as well, but these guys, this guy G is unknown. This is just his transpose. So what does it look like to go from the process of a measured dipolar coupling and an estimated alignment tensor to what G can look like? What does this process look like, right? Remember what we did before is we estimated S, now we're going not the other way, but we're using that information. So the beautiful thing about this is it works because we're in a global coordinate system, that even though I might have estimated the alignment tensor for this helix very far from where vector G is, this bond vector, I can use the alignment tensor determined on this helix, which might be physically far away, to estimate or compute things about the orientation of G. So that's what I want to show you next, how this is used in a system. So with that having been said, let me pause. Feel free to take a break. I will come back in 2 minutes with some tea and I'll show you some slides. All right, hello everyone, welcome back. If you had a chance to get something to wet your whistle also. So I'd like to show you some, some pictures that I made that I think will answer some questions and help you visualize some of the things that are hard to draw. So let me share my screen and we can look at this. All right, tell me if you can see that.
Yep.
All right, excellent. All right, so I want to show you some pictures. So I think it'll make some of these things a little clearer and also be kind of fun to visualize. They're really quite interesting objects. And they're based on chapters from the textbook, as you can see. All right. All right, so once again, we're looking at a lot of details here. So just to kind of motivate this, here's a molecule we showed last time as kind of a warm up. And, these tiny changes here can make a lot of difference. So, here's the difference between theobromine and caffeine. Does anyone know what substance did I have the last 5 nights that has theobromine in it? Does anyone know?
Chocolate.
Chocolate, very good. So, also usually has some caffeine as well. So these tiny differences we're making in these chemical species and decorations on proteins and other molecules are super important to be aware of. Okay, so, the viewpoint of this course is we want to understand these differences, but also to solve structures and rationalize the mechanism of action. So in addition to saying what happens with the inhibitor itself, we can say things like, well, what's the structure of the receptor that binds the caffeine and theobromine molecules here, right? If you look at the differences here. So this is a very nice study of this receptor. So this is kind of what we're trying to get out of this course. How do we get at these structures, understand them, and design new things? All right, a membrane protein too. So this is a challenging project. All right, so again, thinking about this, we looking at two big themes of this course. Technically, the third big theme, of course, is looking at things related to SARS-CoV-2, which we'll start very shortly. But again, this is part of the theme where we're figuring out what is the structure of biomolecules, such as macromolecules like proteins, RNA, DNA. So how can we go from that to measurements that are indirect, do a lot of computation typically to try to determine a structure and have that be robust and correct. So we're in that theme here. And again, I want to mention these can be really complicated. So this was some work done by my student now Dr. John McMaster in my lab. This is 550 amino acids, 65 kilodaltons, one of the largest proteins to be investigated monomerically by NMR. And as you can see from this modified HSQ spectrum, a lot of different spots, one corresponding to each amide resonance. If this had been decoupled, we'd see 4 times as many spots. So lots of reasons to do that. Just to give you an idea what these spectra look like in the wild. And I'll be presenting some work done by collaborators and friends, some of whom you're reading too. All right, so let me try to motivate this by contrasting and answering some question about how traditionally, what are different pathways through the NMR process. So as we've been reading about, you might do toxine-cozy experiments to determine through bond connectivity and assign the backbone, even assign the side chain resonances, the backbone resonances, and you have to assign everything. You have to have every spot assigned to a tuple of atoms. For the HSQC, it's a pair. For 3D nosy, it's a heavy atom and 2 protons. For HNCOCACB, it's 3 backbone atoms that are in a distinguished subgraph. And then people typically use a form of simulated annealing. It's not really molecular dynamics, it's MD engine, but it's not proper MD because it's simplified. And you get what's hoped to be a reliable initial fold. Then you assign the NOEs based on the structure. In other words, you say this was my structure, so now I can assign the spots. Then you do a full simulated annealing protocol and refine the structure. So RDCs are typically only used traditionally at the very end. And this is because they were discovered much later than the other kinds of data. And moreover, their energy landscapes of these things is very rugged as conformation changes because they're so sensitive to geometry, namely quadratically sensitive to changes in orientation. So there are many issues we'd like to improve in this, and this is a great place for methodology people. So one is it's hard to do assignments. You have to have nearly complete assignments in order to do this, nearly 90%. And the initial fold is made only using NOEs. So we're going to get some possible errors there. We're using a subset of the data. It's not globally referenced. Simulated annealing, molecular dynamics frequently get trapped in local minima as they're used here. So you're not going to get optimal solutions. And that's going to then give you, in this case, misassignments sometimes. And lots of manual intervention is needed in these structures when they're complicated. So not at all a turnkey process, although many structures have been solved. So since RDCs are so interesting, is it possible to use them earlier in the process? And here's one possible pathway, right? The pathway would be something like use a technique to define, calculate the structures of the secondary structure elements, helices and strands, right? And then once these are calculated, you not only have the structure, but you have their orientations. So even if helix 1 and helix 2 are super far away from each other in the protein, like even tens of angstroms, the orientation will be correct. So then it takes relatively few NOEs to pack these together because you're not simultaneously resolving the orientations. So it takes a fair number of NOEs to pack together 2 more or less rigid elements if you don't know the relative orientations. But if you know the relative orientations, it takes 3 or 4. Then you can pack them together. Once you pack them together, you can then use various protocols, which we haven't talked about yet, which are very interesting, that try to use RDC data and NOE data plus kinematics to model the loop structures on these. And then you can, from that information, you then can assign based on the structure, the side chain resonances and complete the NMR structure determination process. So this inverts what we saw before. Rather than using RDCs at the end to sort of refine things and tune them up a little bit, we use it in the beginning because they're globally referenced and in some sense one of the most reliable measurements. So we try to drive things by the measurements of the RDCs. Now, when this was first proposed, it had the term exact because mathematically to determine these structures, you're solving for the roots of polynomial equations in one variable that are of degree 4 or less. And as we know from CS230 and also Galois theory and a lot of algebra, undergraduate level algebra, those equations actually have exact solutions in closed form. And when mathematicians and scientists find exact solutions, like SVD, they prefer them to Monte Carlo or simulated annealing. But the name was changed because biological audiences, when they see the word exact, sometimes thought that exact meant biologically exact, i.e. exactly what happens in vivo or in vitro. And so the data is from Peugeot's lab, and I'll use this as an example here. So unknown before, now it's solved using these techniques. And one thing that happened while this was being solved is that the X-ray structure. Oh, did the audio stop? Did we have a problem?
Just very briefly, I think.
Are we doing okay now? Did you miss anything important that I was saying?
I don't think so. Thanks.
All right, thanks. Yes, please do let me know. All right. So here's a kind of model protein. One thing that was interesting in this case is that the X-ray structure was solved by a structural genomics initiative around the same time, independently. And so by the time the papers came out, we can compare the NMR structure with RDCs to the X-ray structure. And there, of course, we have statistics on this like RMSD, but they actually line up pretty well. So there's reason, proof of concept that such a thing can actually work meaningfully and also be mathematically elegant. There's also was at some point an NMR reference structure solved, so fairly similar to traditional NMR methods, but using a bit less data. So good correspondence with orthogonal methods, either in terms of biophysical techniques or in terms of algorithm. So how could we do this? So basically there's a few sort of key ingredients for thinking about this. So one is RDCs, which we talked about. And the key way to think about this is that the RDCs are going to give us constraint on the geometry of these vectors. And I want to talk about what that geometry actually looks like. Basically, once you have determined the alignment tensor, and when you have an RDC measurement, then the vector that you're interrogating is confined by that data to lie on one of two curves. And this corresponds again to the symmetry of the dipolar operator, top curve or bottom curve, looking something like this. So the data from one RDC measurement on one bond vector, when you know the alignment tensor, means that that bond vector is lying on somewhere on this curve or this curve, you don't know which one. And these aren't always circles. They're sometimes called Pringles after the potato chip. They have a sort of funny shape. But the reason this is kind of interesting is that if you record RDCs in more than one medium, or if you record more than one type of RDC, for example, not just NH, but also C alpha, H alpha, you combine these into analytical or really exact solutions. So in other words, you can attain exact solutions to the orientations of amide bond vectors and what we call the CH bond vectors, the C alpha to H alpha orientations, and by kinematics, the phi psi angles. And this is done by solving an equation that is merely quartic. Therefore, it has exact solutions, fourth degree. The U is equal to a tangent of a half angle. And then we can also use some tricks from a branch of mathematics called algebraic geometry, now very popular in computer science. So, I mean, it's very weird if I were to say, you can't say using traditional algebra, I want the minimum of some function. That's a very difficult thing. But if your function is algebraic, you can cast it like this. This is a strange way of casting minimum. So I can say there exists an X star such that for all X, f of X star is less than f of X, then there are algorithms from this field of applied algebra, basically, or algebraic geometry, that can then solve for this optimal solution because you've recast the minimization problem in a way the algorithms can deal with. So basically 3 tools. The geometry I'm starting to develop, the fact that polynomial solutions have a finite number of roots and we can solve them exactly, plus some very elementary techniques from basically algebra that do optimize geometry. So let's talk about this. So here's a picture of the geometry of a piece of a protein. And notice I have the phi and psi angles here. And I have all these bond vectors. Here's various bond vectors on which I can measure RDCs. So obviously I have amide bond vector here. I have my CH bond vector here. I can also measure it on C alpha, C beta, and I can measure C alpha carbonyl. And I can even measure it on carbonyl nitrogen and even carbonyl carbon to amide proton, but this will depend on the orientation of the phi bond angle as well. So I'm kind of a little more reluctant to do that. No, sorry, this will be fixed in the peptide plane. Sorry, some of these will be, these are all invariant under angle. So the peptide plane is here. These are all more or less invariant under angle. So there are different properties of how efficiently I can measure these guys, but they're all things that in principle can be measured, although the amide bond vector RDCs are the easiest. Probably after that, the C alpha, H alpha are the easiest, but sometimes hard to get at. And they can all be measured in one experiment. That's pretty cool. Obviously, you need C13 labeling to measure these carbon ones. So I can measure these and I can now put them together in some kind of framework. So again, here's our framework. I'll review for what we had before. We have our protein. We put it in an aligning media here. Bicelles, micelles, phage, stretch it into gel. Orientation of a bond vector relative to the external magnetic field. That's the theta here. Here's the equation that describes dipolar coupling. Using the simple derivation in chapter 5, we can turn that into our tensor format here. The dipolar coupling is equal to a constant times V transpose SV. V is a 3x1 vector corresponding to the XYZ coordinates of a bond vector. S again is a matrix which is 3x3 real valued, traceless, and symmetric that corresponds to the orientation. And again, you can visualize these in certain ways. By SVD, we can decompose this into a rotation matrix called a principal order frame and a diagonal matrix of eigenvalues saying how it is stretched. So again, we visualize this U as being a kind of rotation, and then the eigenvalues is stretching it just as we did in PCA. So the key thing here is the measurements are global. If I know, look here, if I know D, D max is a constant, we know S, then if I ask what's the orbit of V's that solve this equation, they look like this. They look like these curves on the sphere. And here's an example here done by my student Tony Yan, has a PhD many years ago. But if you fix S and you just vary D and you ask what the curves would be, this shows how the curves, solution curves for the vector V change as I change my dipolar coupling. So a very interesting structure. And each of these curves actually is the intersection of the two-dimensional sphere with what's called a spheral conic. Okay, so this is pretty neat. Now, one thing I can do is I can measure the same bond vector in 2 different aligning media. So for example, phage and bicell, or stretch gel and phage, stretch gel and bicell, so forth. And if when I do that, I get different principal order frames, i.e. this matrix U here is different, then I get 2 different independent measurements. And that corresponds to a constraint that my vector has to lie on 2 different curves on the sphere, drawn like this. So the yellow curve is from one experiment, one RDC experiment, one medium, and the orange curve here is from a different medium. And we know we have to lie on both, so therefore we have to rely on their intersections. So we intersect these 2 curves and we get a finite number of points. So this is the intuition for why there are exact solutions. Namely, we solve for we compute the alignment tensors for both the yellow and the orange medium. Each one then gives us these curves, which are algebraic. We take the mutual simultaneous solution or intersection of those curves. We can calculate these points exactly in closed form. Then we have a choice depending on which branch. In this case, we would have a choice of 4 possibilities. And so as we march down the peptide plane, making our geometry, we have a branching tree with a branching factor of either 0, 2, 4, 6, or 8. So RDCs and 2 media can be used to pin down my solutions so they're not on a curve, but actually are points, which is quite remarkable. So one question is, many experiments just record 1 RDC per residue. Like 1 RDC per residue, is that enough? Can you define a protein backbone? Let's investigate this. This is a question of, is the information sufficient? It's very different from things you'll see sometimes in biology and other fields where they just run a lot of experiments, computational experiments. We're trying to mathematically characterize if the algorithm is sufficient to use that information to calculate a unique structure. How can we investigate this? So again, we're going to align the protein in some medium like this, and we're going to basically, first thing we're going to do is estimate the alignment tensor. So again, each RDC is equal to a constant V transpose SV. V is the bond vector, S is this SOAP matrix or alignment tensor here. You can visualize each of these as a sort of rotation matrix, but once you know S, if you've measured R, this constrains V to lie on these curves here drawn in orange. And in order for this approach to work, you have to calculate S. So you have to know it, but then it's global and it only has 5 degrees of freedom, as I indicated before. Why 5? It's 3 by 3, but it's traceless. So one element of the diagonal depends on the other 2 elements. And it's symmetric, meaning the off-diagonal elements are pairwise identical. So you only need 5 degrees of freedom to do this. Okay. Now, often this is done in terms of direction cosines in the literature. I don't really like direction cosines. Physicists like these. You can kind of think of them this way, but I'm just going to use things like dot products instead. But basically, the idea of this construction here is when we go back to our equation here, what this does basically is diagonalizes the alignment tensor. So S is an arbitrary 3 by 3 matrix that's traceless and symmetric. When I use SVD like this, I have a rotation matrix here and it's transposed. Sigma is basically a matrix which is diagonal, right? So that's called the diagonalization, this process diagonalization of S. So what does that look like when I write it out as scalars? Well, it looks like something like this. So when I've diagonalized it, when I've moved to the principal order frame by doing a change of basis, namely rotating by R, I can write my dipolar coupling just like this. These are the diagonal elements, S, X, X, Y, Y, and Z, Z, times my vector, and it's squared because of the symmetry of the dipolar operators. That's my RDC equation once I've moved to the frame that's sitting on the molecule. So things look like that. So already it's looking like an algebraic equation, which is pretty neat. Okay. Now, so that sort of tells me something about 1 RDC per residue. We don't really have a lot of information. We know that each bond vector is on a curve like this, right? So that's kind of interesting. So remember our original question here, can 1 RDC per residue define a protein backbone? So like just amide, proton, just CH. And the answer is basically not quite enough because it defines each amide vector is going to be on one of 2 curves like this, but it doesn't tell you where you are on the curve. So what about 2? How about 2 RDCs per residue? That would be pretty interesting. So now we're changing the amount of data. We're asking if 2 RDCs per residue can define a protein backbone. So again, think of our situation. We have the phi bond angle, the psi bond angle here like this. We're going to try measuring the NH RDC and the CH or C alpha H alpha RDC and seeing how these work. So the key thing here can be thought of as follows, is that if I look at my CH, imagine I've sort of do this by induction. So imagine I've done the peptide structure from the left, right? And so in particular, I know this peptide plane here, and I know the atoms in it like this. So I know this guy here, and what I'm imagining I'm doing is I'd like to then measure this CH RDC and use that to kind of continue the chain like this. So if this is all fixed and known, the unknown variable here is the phi bond angle here, which can rotate around. And the idea is, does the RDC report on what that value could be so I can solve for it? So basically, the way to think about this is if you imagine the orbit of this bond vector, as I rotate phi, it's in general going to be an ellipse. In many cases, it will be a circle, but let's just say an ellipse. So what I can do is I can write down the possible orientations of this vector as simply a function of my phi bind angle. Now I have some constants in here like phi 0 and A and B. They're just constants dependent on the kinematics. And as I change my free variable phi for residue I, the idea is this vector rotates around like this, and it describes some sort of curve, let's say ellipse in this case here, and the CH vector has to align on that. So this is purely from kinematics. It's known just from modeling of the peptide plane. There's no data involved here, except for our knowledge of how proteins work. So that's the kinematic ellipse. And now we're going to intersect that with the data. So from what we had before, the RDC for the CH bond vector is equal to this. Again, this is the diagonal of the alignment tensor. X, Y, and Z are the coordinates of the CH bond vector. So now I have the green ellipse from modeling from kinematics of what proteins look like, peptide geometry. I have the orange ellipse or Pringle from the data. Now I just intersect these guys. They have a finite number of intersections. So I pick one, or I do a branching search here and move on. So now what I can do is I have an equation that basically says that, I mean, I can't directly operate on the angles, but I can operate on cosines of the angles. This is like rationalization, right? So if I have a complicated equation that has sines and cosines, I can substitute C for cosine and S for sine with the added constraint that S squared plus C squared equals 1. So now I have an algebraic equation in basically the three-dimensional coordinates of this CH bond vector, the rotation matrix that took me in here that defines this peptide plane, and the cosine of the angle phi that has to equal 0. So this algebraic equation, I can use that to solve for cosine basically. And then I can move ahead. So once I've chosen or have a branching search to choose the CH bond vector based on intersecting the data with the kinematics, I can now fix that solution and move on and ask the same exact question, transpose, so to speak, about the NH bond vector. So here I have a free psi angle here. As it rotates around, this is all rigid, but this thing rotates with psi, again, along some ellipse. I can write down an algebraic equation I have to solve that there too. And it will also have finite solutions. And all these equations are derived in chapter 18. So in short, in this case, we have the kinematics of the CH and NH bond vectors derived from ideal peptide geometry modeling. And we have the data telling us on what curve the CH bond vector or NH bond vector has to lie. And then we have the rotation matrix, which is our induction that takes us from how we came before. We combine those and we chain forward to solve for the structure. So this says that in principle, we can compute essentially all backbone dihedral angles based on a good measurement of NH and CH RDCs, almost, right? You have some things. So we can have a theorem. You have to know things, the bond length, bond angles. These are the non-dihedral things, the peptide plane dihedral angle, the alignment tensor, and then you can compute the set of, not just one, but the set of all conformations consistent with 2 RDCs per residue. This set is finite and algebraic, meaning you can compute it and you can score it. This is a very unusual theorem. Usually in biophysics, it's much more difficult. So this tells us that this is almost enough data to solve a protein structure ab initio from exact equations. And that's, I think, really interesting to our class because we're interested in algorithms we can understand the complexity, completeness of, and so forth, just using geometry. So that's why I think it was worth going into this geometric analysis here. All right. So
a quick question.
Yeah, absolutely.
Yeah. So if we're doing this kind of inductive process where the solutions to later, each one depends on the solution to the one before it, don't we kind of lose some of the benefit of having globally independently defined RDCs where errors won't correlate and build up?
Errors can't build up because the orange ellipses are fixed. They don't change, right? So this ellipse is always the same. It's not influenced by anything previously. It's always globally referenced.
So wouldn't the green ellipse change if we're depending on what the orientation of the previous peptide plane was?
Well, there are only 4 solutions for that. So you look at all 4, right? You don't just choose the best one. That would be a greedy search. So what you do is you have a search that branches has a branching factor of 4 at each one, and then you're going to, I mean, you could explore them all, but you probably just explore the best ones, right? So you're not throwing out any solutions, they're just further up in your search tree.
Okay, I guess like what I'm thinking is like say there's an error in one RDC. If you're doing like the one with the two NHs, like that error and wouldn't change the result of like the next RDC or the next residue, but in this case it would.
That one's actually much easier to argue because everything's globally referenced. So if we use, if we use this to resolve the, so we'll have 8 possible solutions, right? So if we use that to resolve the amide bond vector in residue I, and then in residue I+2, that's solved completely independently. It doesn't use any information at all from other residues. So you're just fixing them and you're finding kinematic solutions between them. So there's no problem. In the case of 2 different order frames to pin down amide bond vectors, there's actually no information propagated between the residues at all.
Yeah, that's what I'm saying. So like, to me, that would say like that would be way better than like the NHCH one where you do have information propagation.
Way better. Well, it is more information. I mean, 2 media is more information, right? But I don't see an argument that it's way better. Like it's a different setup. So in practice, you still have these things. I mean, it's not clear how a previous choice contaminates this unless it contaminates the discrete choice, right? That's a possibility, I suppose. But we don't have any data to show that it's actually worse. I mean, protein kinematics is pretty reliable in some sense, right? So I don't see a problem there because proteins typically don't do things other than this. So I think it's a theoretical argument that can happen, but in practice, there doesn't seem to be a problem because things get pulled back to reasonable solutions by the yellow solution. So you'd have to have a lot of things conspire in order for that situation to be a problem. But I think that a detailed study of which is better is, something that is kind of pending. Like all we know is that they both tend to work. So that would be interesting to do something that really tries to quantitate that. But I don't see an a priori argument that one would have to be better. I see sort of hypothesis that one might be more reasonable. I think that the thing that agrees with what you're saying is that if you had 2 media, why not record NH and CH in 2 media? Like that's clearly going to be better than just two things. But we're asking here for the minimal information requirements if the data is good. Does that make sense?
Yeah. Okay. Thank you.
Good. Interesting question. Okay. Yeah. Okay. So where we were was we kind of picture of the search, right? So basically in each peptide plane or each bond vector, right? What we're doing is we're intersecting a kinematic ellipse with an ellipse from the data to get discrete solutions for the bond vector. And then we're combining these. We have sort of a large search for these things, and it can be rather large, right? So this is why, you need some algorithms that can deal with this. So typically what's done is some kind of scoring system along the way. So you take the top things. Some of these solutions will be kinematically plausible, but not biophysically plausible. They'll cause steric clashes or be terribly far from the Ramachandran diagram. So typically you can prune this down quite a bit. But it's kind of remarkable that the data actually gives you a discrete search. So what we've done is we've taken this problem of solving protein structure, and either in the 2NH case or in 2 media or the NH plus CH case in a single medium, we've taken that problem and we've reduced it to a merely discrete and finite search, which is, I think, pretty exciting because then we can bring together the apparatus of discrete mathematics and optimization algorithms to try to solve it. So how would you actually implement this? I think this gets at some questions we might have. So way too many things to explore them all. So typically what you would do is you'd have a confirmation tree where you consider these different solutions, right? So basically you can prune out some of these as being Ramachandran outliers or being steric clashes. What often does impossible in practice here is when you look at this, we have a particular RDC value, but what you might do is you might throw a distribution around that, actually sample the RDC error interval. We have very good ways of calculating what the error interval is for RDC. So typically, for example, 1.1 hertz or so for NH and somewhat larger for CH. So you could sample that interval and run each of those discrete calculations in, and you get a series or an ensemble of confirmations instead. So this is a search that's like the searches Andy's been teaching you, that we used to have a search tree. You can do A star or best first or a variety of calculations here. It's usually important to keep other solutions and backtrack, but the idea is that you would try to choose solutions that were optimal with respect to Ramachandran outliers, steric clashes, energetic considerations as you move through this, and get the best solutions that are actually feasible. And then many solutions will be pruned because their steric clashes are outside Ramachandran regions instead. So in this sense, the algorithms keeping track of alternative possibilities, you could easily have something that looked really, decent up top and got pruned later. So that's how you would search such a big thing. We can talk more about that. And finally, just a few other things, and then I want to take some time for questions. Once we've found the structures of secondary structure elements like helices or strands, we typically want to use NOEs to pack them together. This is typically done in a way that looks at a couple of different criteria. So you might look at just the how well do you satisfy the NOEs, and also look at some kind of packing score that just looks at, for example, van der Waals. So how well do these things pack together, and how well do they satisfy the data? One thing that's a little curious about this is that because of the symmetry of the dipolar operator, you actually have ambiguity, although it's discrete ambiguity, in the orientation of these secondary structure elements. So in these cases, and it depends on what experiments you do and what media, but if you have only one medium, say you looked at CH and NH in one medium, then this helix actually has four different, has two different orientational possibilities. So there are four ways to put these things together. So in other words, this has an up or down, this has an up or down. And so your ways of putting these things together are up, 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 down, down, up, and down, down. So in doing the structure calculation, you have another fourfold complication. You have to consider all four possibilities in putting these together. And by the way, these ambiguities in some of these equations were actually pioneered by our friend and colleague Hashim al-Hashimi when he was a graduate student with James Prestigard, worked out some of these equations and some of the techniques for dealing with symmetry, which were very elegant, particularly that he was the first to think about those issues. Okay, so in short, there are sort of ways of packing some of these things together. We've only really looked at a piece here, which is, can we determine these secondary structure elements like helices and strands? We've sort of seen there might be a way to pack these together by searching with a bicriterion optimization over things that try to match the data and try to match the packing. And then we need to do something else, which won't be on this lecture, how to put on the loops, and then how to use those structures to assign the rest of the NOEs, especially in side chains that stick out. You could simultaneously assign the resonances on the side chains and also assign the rotamers using these techniques as well. Okay, so let's look at just a few examples, especially for the experimentalists, and I'll try to maybe leave 15 minutes per question. So, this has been tried on a number of proteins, both known and unknown. Let's look at some of the data, how this looks, how's the result, and give you an idea of this. So you might use different data for this, like some NOEs, RDCs, and so forth, what we're comparing to. And then what kind of accuracy do we get? We can look at accuracy of the secondary structure elements. This is really what comes directly from the RDCs. And then look at accuracy of the actual backbone, like how close did we get to the actual backbones when doing techniques like this. So reasonably close in terms of these. I mean, below 1 angstrom is really quite good for these kind of structures. Here's a way of drawing this issue. So one thing that's beautiful about RDC data is that you can simulate it from a structure. So if we go back to our, I'm actually do this, go back to our equation. Let's see now. So if we have a structure, then we know all the V's here. And if we have an alignment tensor, then we can simulate or back calculate all the dipolar couplings. So that means for a structure, you can calculate what the RDC data should be if your alignment tensor is correct, right? So that's called back calculation. I call it simulation, which is pretty reasonable, I think. So that means that when we have our data, we can then look at this data to show the correlation between the back computed and experimental RDCs, right? And these should have a good correlation if things works. And then we compute confusingly an RMSD between these values, not structural RMSD. It's in units of hertz because it's measuring how close was your data. So in this case here, we can see that the agreement is really quite good. It tends to be worse for CH because it's a noisier computation, but it's getting really quite good for these different systems here. So that in other words, when you build a structural model, it corresponds well to the actual data. So again, experimental RDCs versus back computed RDCs here. Now we can also measure how well can we pack these things together. So this is one of the reasons, for my claim that, we get good fits using any of these techniques. I mean, this technique really is using essentially NH and CH in 1 or 2 media. 2 media is better if we can do it. Again, this is for a new protein FF2, again, rather good. So you can see sort of the errors here. NH is the best, CH is pretty good. The signals are weaker for C alpha carbonyl and for nitrogen carbonyl, so the fit isn't as good. This is pretty typical in this area. So you can use this data, but it tends not to be quite as accurate. So this is a case of actually using 4 RDCs per residue. Now, we can also try to have systematic algorithms that pack these things together. It's worth looking at how well those algorithms do. I'll just show you one example here. Typically, you get out these ensembles where you're scoring these not only by how well do you satisfy the data, but how well do they pack based on a model. And you're pulling out structures from these. So this is basically packing together these secondary structure elements, each of which has been determined by looking at exact solutions to RDCs, and then using NOEs to pack them together without even having the loops in here, right? So we can sort of combine these. I think I'm not going to show you all of these details. Let's look at just a couple here. We can combine these and get final structures. So again, here's an example of packed structures in this one case here, agreement with reference structures. So compared to the X-ray structure, it looks pretty good. The statistics are good. There's an NMR structure also looks quite good when we do that. So basically, this is a case of just using CH and NH RDCs and only sparse NOEs that are relatively easy to assign. So again, many cases where we can look at this, they seem pretty decent. Maybe I won't go through each of these. You could again superpose these and look at these again. Now there's some tricky things if you want to pack together beta sheets. I'm going to let you read about that, but basically you have to augment this protocol to take into account the structure of beta sheets because you have to take the geometry into account as you're growing these. That can also be done. Basically, you imagine computing one strand and then having the others build out from this. I think I'm not going to cover that here in interest of times. So in short, to kind of summarize where we are, traditionally RDCs are used at the end of structure determination. So the idea was, can we try to find a way to get around these problems of propagating errors, local minimum, manual intervention, by using the RDCs first? This is kind of an RDC first approach. So I think that it might not be the final way that people use RDCs, but I think that it's an interesting attempt to use exact solutions, globally referenced data, to drive the structure determination process to make it a bit more like crystallography, which is globally referenced. So I think that's sort of what's really driving this. So there should be some advantages to doing this, and I think it's still a research area about how to do this correctly. And there are many parts of the algorithm downstream that we haven't really gone into yet, but we might later in some of the papers or in the class. And again, on reference cases, compared to both X-ray and compared to crystallography, these structures tend to be pretty decent. So you can both do this prospectively and retrospectively and get very similar structures using less data and referencing it to global information. But I think from a kind of computer science point of view, as we go through this, or computational biology point of view, instead of it being a big heuristic stochastic optimization process by simulated annealing, we can understand the geometry well enough to say that the solution structures are actually something we can compute by exact solutions. And then we can use those exact solutions in some kind of discrete search in order to enumerate, if necessary, all the structures, but certainly the best structures. So I guess the analogy I would make is that we know physics can be useful because it gives us equations. When those equations are exact, we can use them to solve precisely what's the answer. So, no one would try to solve, if I throw a ball in the air, where would it land, using simulated annealing, Monte Carlo, or genetic algorithms. Rather, we will write down the equation for a parabola and see where it hits the ground. And you can solve that exactly by solving quadratic equations. Here we're solving quartic equations, but there are many cases where actually the equations are somewhat simpler. We have cases where it's only cubics or even quadratics. And I think that if you meditate on it and realize that there are many situations in NMR, whereas if you collect your data, you can solve the structures by a series of linear equations followed by a series of quadratic equations, that's kind of reassuring that we're understanding the process and we can really get excellent solutions based on the exact solutions to these algebraic equations. Again, there's more data. I'll put these on the web here. Lots of references to look at if you wanted to explore this area. So I would say that some of the lessons of this are that RDCs not only define structure, but they actually help assignments. Because if you can define this structure, and if you have an NOE you're attempting to assign, you could see if that distance restraint is compatible with your structure. So I think this is a nice insight. It's well known that structure can help with NOE assignments, but the RDCs directly deriving this is kind of nice because then your assignments are globally referenced. So again, I think that the protocol that might make sense here is to start with a global fold that's calculated from the exact solutions to these RDC equations. We can apply a systematic search to pack them together using easy to assign sparse NOEs. Then we can use that to put on loops and then resolve the rest of the NOE assignment problem. So I think there are a number of things to be said for it, but from our point of view in this lecture, it's the information content in RDCs in some sense. And again, this is a collaboration. A lot of the data is from Peugeot's lab, and other people have been able to contribute to this, well, mostly in my lab as well. So with that, I want to leave some time for discussion. I want to stop sharing the screen and ask if you have any questions or anything you'd like to talk about. I'd be happy to talk to you. And hopefully this was interesting and useful to you. Any discussion? Any questions? So one thing I wanted to say is that I think when these experiments were discovered, I think people were very happy to put these techniques into their kind of existing packages. And so it's my impression that the use of analytic techniques and exact solutions is kind of a young field. So many interesting things can be done from the point of view of people looking for, projects or PhD dissertations or, applications of research, and they should be applicable to cases where the systems are very difficult and we can only get a small amount of data, or that we want to be very certain of what we have despite the data being sparse. That's what I think is the payoff for these kind of algorithms. There's not only are they, I think, useful out of the box, but I think there's lots of great things that can be done by young and old people who want to find computational problems and apply them in this area for solving biological structures and characterizing their dynamics. Yeah, so let's look at beta sheets a little bit. So there are several ways of looking at this, both from kind of a history of science point of view and point of view of what's interesting to do kind of next. So one way is that, well, there are all these tools, right? And we observe these issues when just trying to apply them out of the box to beta sheets. And so then it was, okay, well, now we need to, find some way to accurately compute the beta sheets. So all the theory works fine here, but there were some difficulties. So first, it's not always true, but some of the strands are very short. And when they're short, it makes it somewhat more tricky to and if those bond vectors are actually parallel, then the system drops in rank and is somewhat degenerate. So when you have such a small number of bond vectors, it can be that you're it can turn out that you are going to have difficulty in computing accurately the alignment tensor. So maybe I'll share my screen and you can see some pictures of this. So I wanted to give you a little way of thinking about it. Let me know if you can see this. All right, does that show up for you? Can you see the screen?
Yep.
Yes.
Okay, good. Right. So again, one of the issues is that that if these strands are short, it's somewhat more difficult to estimate the alignment tensor because you need at least 5 independent vectors. So theoretically, they're kind of there, but if you had more, it would be more accurate. And they're typically not parallel; they typically alternate up and down. Sometimes in beta sheets, at least in our systems, you could have missing data. You could have some uncertainties in the RDCs and the NOEs. So missing data can be sort of important here. And basically, if you can have NOEs between the beta strands, but it turns out that if you are trying to simultaneously assign if you're trying to assign the rotamers, basically so I guess the way to think about it here is that here we have an example of an NOE between one of the H gamma protons of valine 5 and one of the H delta protons of isoleucine 13. So we might know something about that NOE, but we haven't yet assigned the rotamer. So if these rotamers can really be, can really be different positions, that can change the packing here in some sense. And so we actually went to a scheme that was more complicated experimentally. So first of all, how would you even get unambiguous sidechain NOEs? Well, we have to go to a scheme where we methyl label only the isoleucine, leucine, and valine. And then you would get NOEs between basically methyl protons and amide protons, right? So it could be methyl methyl, methyl amide, amide methyl, or amide amide. And these are unambiguous, but you still can have rotamers for these. And so that there's some complication in that the distance depends on the rotamer. So you're simultaneously assigning the rotamer and the distance. So in short, can be tricky to estimate the alignment tensors of things that are very short. There tended to be some missing data. It tended to impact these a bit more than some other structures. And there's an interaction between packing the two strands based on a distance when the distance depends on the assignment to a rotamer library, or a rotamer in a rotamer library that you're assigning at the same time, you have some choices there. And this is even true when we used a more complicated labeling scheme, a very nice labeling scheme that at least made these unambiguous as to which proton they were or which nuclei were involved, but made it somewhat more complicated. So in order to get around that, we have some ideas, and one is basically that might start with one strand, the strand with the best data, right? So compute this strand in the same way, but then as you start packing other things around it, so you pack this one, it says, as you pack the other things around it, what you do is you do an approach to creating this where you're simultaneously satisfying the RDC data and the Ramachandran and the sterics, but you're also satisfying how it can build out to the other strand and satisfy the NOEs. So it basically, you're packing the strands while you compute them. So in other words, instead of being able, if you might imagine at each, if you have 4 to 8 different solutions for each phi or psi bond angle, you can imagine this strand potentially going off in some crazy direction like this. But if it has to pack against an adjacent strand, that can be used to prune these restraints. So basically, as it goes down, as the second strand goes down in some sense, like this, what it does is it's going to have part of the scoring function for which solutions to pick be based on how well is it satisfying the interstrand NOEs, which are unambiguous. So that was kind of an interesting approach. We were able to get out decent solutions there. So we're basically taking into account the beta sheet geometry in order to extend this. So I guess one way to think about this is, suppose, think of it as a project. Suppose you're working after someone in a laboratory doing this, or you're looking for a course project, and it's been shown that you can pretty easily compute secondary structure elements consisting of helices, and you could actually compute some strands, but packing them together tend to be more complicated. What do we need to kind of add to the algorithmic process? So basically, the scoring function that ended up being used here, again, you have exact solutions, so you're finding a way to score the exact solutions. That's what we're thinking about. So exact solutions still come from intersecting these either data curves or data and kinematic curves. But then when you get all these different discrete combinatorial solutions, how do you score them? So you look at overall RDC RMSD, so how well does the back calculated and the experimental RDC data fit? How far are you off from standard strand geometry? So we know what beta strands should look like. We can have them bend a little bit, but that's, we want to kind of penalize that. We know the pattern of hydrogen bonding that should occur in strands. We know the pattern of hydrogen bonds for both anti-parallel and parallel. Beta sheets, that was actually used explicitly in the jigsaw chapter that you read for NMR assignment. So we want to score that, basically we want to reward the formation of expected hydrogen bonds and penalize when the hydrogen bonds can't be made. And of course, as before, we have the NOE RMSD, that is the closest for the distance restraints. And to check for steric clashes, we have an energy function there. So we have the linear combination of these things. There's some fitting, and we minimize that. And then, of course, we have some experimental results on how to do it. So here are 3 proteins, ubiquitin, with 2 different kinds of data, right? This is kind of interesting in some sense. And here's Dini over here like this. So basically, this is, interesting setup in some ways. We could sort of look at this and also calculate how close we could get in calculating these strands. So, it seems like a promising approach when you incorporate a bit more data to calculate these structures. So that's kind of what I wanted to get at for you. You have to add some things in order to put this in a framework where you can actually reliably calculate beta strands as well. Does that make sense?
You mentioned like that with only 5 residues, sometimes the shorter ones, it's hard to compute like the alignment matrix, or the alignment tensor. But do you have to compute the alignment tensor for every substructure, or just once for the entire protein?
Yeah, this is a great question.